right. You guys ready? Yeah. So this week is over. The time of the red text has begun. Do this. Don't worry. I think you'll be all right. So, cells, beginning. This is red text, and I have started recording. This will stay up, obviously, just out of convenience. Hooray for us. I am unmuted, unlike for genetics yesterday. I forgot to unmute, whatever. Dang it. Good thing it's not live. So, on our sugar conversation, what is the target of a drug? This is actually a good discussion. Technically, there must be some involvement with disease. And hopefully the target has nothing else to do with other good stuff. You imagine drugging something like something that's involved in like all your processes, and like then the body goes crazy and dies. It's gonna be horrible. So you're looking for something specific for a target. I'm gonna write that for us too. Something specific. Next, hopefully your drug mainly runs around the area of disease, should there be a local area. Notice how the theme here is sort of sparing the healthy stuff in favor of really getting after the bad. Makes sense, right? So again, you could even use the word specific one more time. Last thing, that target has to be at least accessible. You have to actually get your molecule, or we'll meet a lot of other stuff that's not just organic molecules here. Got to be able to bind and do something to the thing. This is one of the challenges that we've had with cancer medicine. A lot of bad actors in cancer cells are very far in the nucleus. It's very hard to get things through there. The other idea with point number 3, the access, is think about all the issues with brain drugs and medicine, right? You got to get past the brain barrier. And that thing was built to not allow foreign entities to go past. So, with the finale, as broad as it is, very few substances that technically do not manifest some kind of change in your body in any way, like we saw with sugar. Technically, that's not only getting metabolized, it's binding to targets, activating them, doing stuff. In the beginning, lots of stuff still comes from plants, right? Poor plants are complicated, so we don't study them, especially not in this room as biomeds. We just got scared. But look at all these things. Think how they've affected history or trade along, across the years and stuff, right? Look at digitalis in the lower right. So it's essentially the red text here. And so yeah, for those of you that didn't have the title, if it's red, but it's like a lot of stuff, main idea type stuff. Yeah, I'll kind of help and adjust the map. But yeah, digitalis is a freaking plant poison from foxglove, right? Poor people that tried all these plants the first time were like, oh, let's see if this works. He's dead. But his heart condition stopped for a bit. It's a long trial, and you gotta be honest, before the days of clinical trials, which we'll go over today, this was a bit of a trial and error type of thing. So take a look at our fun one right here, caffeine. Strangely enough, caffeine evolved because it's toxic to insects. It fries their little cardiac system. For us, though, big scary mammals, it blocks where adenosine would travel between neurons. Makes that spot. And actually, I probably even misspoke already. It doesn't take their spot. But what it's going to do is it's going to bind to this receptor and kind of give it a little kink there. Suddenly, the gene no longer looks like it can accept adenosine anymore. So strictly speaking, caffeine doesn't technically give you energy. It just stops the energy sapping circadian rhythm that your body naturally accumulates chemically. Don't worry, I'm obviously going to bring in some 5-hour energy and we're going to examine it. That's different. But the reason this is here in red is like, obviously, caffeine is a drug, technically. It has a target. It elicits a response against the specific target. This doesn't go off and, make your skin shed or anything, right? It's got a very specific idea of what it's doing. It antagonizes that target. Can't work, can't pass adenosine through. You no longer feel the wake of tiredness. Easy. And this is a red one. It could be a blue, although you should probably know how caffeine is working. But plus, it's pretty common. Who doesn't drink coffee yet? Survive, please. Don't do it, because then it's over. I did throughout college, and then I don't want to talk about it. So in the beginning days, drug processing was very, I don't want to say archaic. Still use a lot of these principles. Technically, you got to grow the poppy. You got to like mortar and pestle it. And then typically, even though we're not saying, because we're not building morphine in this case, we're not organically synthesizing it like you guys all learned in Orgo 2, good job. We did actually just have to sort of filter it out, basically. This process, as you can tell, can come with other stuff with it. The filtering process isn't perfect in the way that synthesizing a chemical would be. Perfect, quote unquote. So technically, anytime you get a derivative of a plant, a few things are probably coming with it if it's not synthetic. It's a big reason why something like THC and CBD, they're not acting alone when weed is around. Technically, there's other things in the plant that are coming with that product. Same reason why we have kind of moved from morphine into more of a synthetic type thing. So, fun times as far as looking at a cell. The word emergence is really cool and badass sounding. Your cells are made of things that are not alive in any way. That even counts the mitochondria. They can't live without your cells, even though they are little ancient bacteria running around. But emergence is cool. The system, as it's put together, must matter in one specific shape. So the drugs that we will see until the very end of this class, they are not alive. No matter, we'll see some strange stuff until the very end. We'll finally meet living drugs. They're pretty neat, but takes a while to get there. As promised, the giant eukaryotic cell, which I'm going to take for granite like the rock. It's very solid. That help. Mitochondria, ERs, and such. All of these could be targets for drugs. So actually, that's why I loved Pharm so much. Is for years, we're just learning about these stupid cell structures and making little, here's the city and here's the post-mail type stuff. Now we're actually going to go drug them and hurt them and do stuff, especially if they're bad cells. Now, a lot of the times too, these organelles will be going bad and a drug will have a specific target, right? What did we just talk about is a perfect drug target, specific, right? So if I want to cut down something, if I have something to fix about the lysosomes are not digesting, I will target the drug to those receptors. I won't hurt anything else. But as we explore everything, like I said, I'm definitely taking this for granted that, a eukaryotic cell is pretty familiar territory for you. Again, red text, cells do all this universal stuff. Technically, with what physiology is sort of a giant cell biology. So again, that's why we're doing sort of some red right here. Again, seems a little familiar. In fact, actually, you guys want me to go a little faster here? Give me a thumbs down, up. How you want me to go? Faster is up. Okay. I'm doing okay. I'll speed it up a little bit based on the votes, but be okay. Sweet. So how does this matter for us? Drugs are what we use to fight issues in the cell. The issues of the cell are consequences of something went wrong. The issues of the cell systems of physiology are because some cells are going wrong. All you need for a heart dysrhythmia is a couple SA node cells that are just, something's wrong. And now the rhythm's off. So this whole sequence is an if-then. Another thing, we talked about accessibility of drugs. The most accessible thing on any cell is going to be the receptor on top of it. We have drugs that can go across and do the cross through things. Most of our targets are going to be receptors though. They're right on the surface, they have plenty of roles to do. And if we can change them, we can change the inside of the cell and everything else that it might affect around it. Just like this one right here with nicotine. You open these ones, you allow this transfer to happen. It's not the cell that you actually opened that's going to produce the effect. It's the cell that you've opened that's going to travel to the other cells. That's where the buzz comes from right there. But eventually, if you have too much, they ignore it, they need more. They're used to being bound like that thing. So the beauty of this is that cells are literally speaking a language with each other. Certain cells can speak to one another, others are completely foreign. Most and a lot of these interactions are done right next to each other, just like that. Receptor A binding receptor B, eliciting some sort of effect inside the B cell in this case. This could be activate. This could be a stop signal though too. A lot of the cool stuff that we know about genetics and everything else now, we have plenty of genes in humans and receptors, we have no idea what they touch, do, anything. If you mutate them, stuff goes wrong. So even though, again, we have that giant textbook, there's still very much in sort of that alien-like language of how to control these things. And it's always kind of a cool idea to think about that, I don't know, cells inside you, individually, they are not alive in any way. These interactions are really special. So most of your interactions are going to be receptor on receptor. These are our easiest to drug too. 2 targets. Perfect drug's just going to fit in one of those and stop it, right? Easy. Do you think that this is going to be a small or a bigger effect drug in this case? We're affecting kind of an interaction between 2 cells, kind of like speaking a little bit to each other. Really pretty small here. This is kind of some of your drugs out there, pretty small effects. For example, Advil, you don't want, bunches of stuff happening after you take an Advil, right? You just want some pain relief type stuff. Now, there's plenty of drugs out there though. Who's taking endocrinology? Who's in it? Nice. Oh, it's loud. We're going to do a mini endo in this slide. So if you drug a cell that's responsible for signaling around its neighbors, you're going to affect all of them. If you drug a cell that is responsible for signaling into the bloodstream, like long distance, you're going to do the whole body. One of the good examples, one of the drugs, for example, that's a giant endocrine effector that is probably the most common is going to be any of the birth control cocktails. They are everywhere. That is why when you have a large drug that's affecting everything at once and cells that are commanding multiple cells across the bloodstream, you have multiple effects. As we know, those can elicit different effects in different people. So kind of scaling up as far as drugs. We basically had neighbor on neighbor, and we had immediate area effect. Some cells just spit out molecules just in the immediate area, don't make it to the bloodstream. They tell their neighbors something's going on. And then 3, everything. And the blood, a little special here with blood-brain barrier, but like mainly I'm just after the size and scale here. Neighboring cells, or like no, cell to cell, neighboring cells, and everything. So the more side effects a drug has, consider which type of drug that kind of is. How far does that thing scale? And kind of like I have down in the text. Sometimes we can't actually stop a specific target, but we can sure as hell stop its promoter or the thing that activates it, maybe. So that's why a lot of drug targets are indirect. We may actually end up stopping the cell that would otherwise activate the gene that we want to stop. We can't drug it, maybe it's too far deep in the cell, or maybe it's in the brain. So here's endo in a single little hit. Don't tell Dr. Cohen. So, other fun thing, ligands. Stuff that the cells spit out, we call those ligands typically. They have a partner receptor somewhere out there that they're going to activate. As you can tell with this little guy, if it activates and binds its partner with the amino acids right there, these are all genes, by the way, fun. It will release this little secondary molecule that will go into the cell and start doing something. Notice there's a good 1, 2, 3 step. Ligands are typically pretty large. They can't cross the plasma membrane. For plasma membranes, what is it? Charge, you can't cross. If it's too big, you can't cross. But the way that ligands have to communicate is they can't open every cell up because water would go in and burst everything. So they have a secondary communication system. It's like handing off, it's like, it's like Paul Revere handing off like mail to somebody else and then he takes it. Or it's like, oh, a good one is like the Great Wall, for example. When they would see Mongols coming, they'd light a torch, another torch with light, another torch with light, signal, 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 inside. So what happens when those signals come through? This is a full system of all the slides that we've been seeing. This is one of the like bigger ones. You've got some sort of signal. You've got some sort of receptor. Then things change. Every cell's different in these next steps. Good thing red. So you've got something that can float around. You've got something that can bind that floating thing. You've got something that receptor has two roles. Not only does it attach to the target, but below, inside the cell, it then changes, opens up, and triggers a cascade. There are molecules inside all of your cells right now waiting to be activated. Think about how somebody would react if cocaine did bind its receptor into a cell, right? See? It would elicit all kinds of changes in those target cells that would read that signal. And what we do in medicine, sometimes what we do is we mimic that signaling molecule with a drug. We pretend. Don't have the real thing. And what this signaling cascade does gives us sort of a response right here. It's a little ambiguous right now. Here's what this slide fails to do. There's no way to show the scale of this. That receptor, it may trigger second cascade that will do its own thing. Maybe one of these is triggering 3 other cascades down. Single ligand may activate hundreds of different things in one fell swoop. Those activations, what's kind of down there. This is where we get into genetics. The goal of this is to literally activate and make genes. All those little cartoons right there are proteins. There will be other activations. Think about right now. A good way to visualize this pathway is, for example, you guys turned in the homework today, right? See that fear building up in you? Right there, your brain heard something, you're like dislike. It sent neurotransmitters out to your endocrine system. And there was that sudden, what happened was inside your cells that control your heart, for example, they got activated. They said, oh my God, the fear signal came through, boom, activated a bunch of stuff, triggered some genes to at least open and activate in this case. But the long-term fear stuff, that will get you making genes to survive. So anytime a zebra is running from something, it shuts down, and this will also go the other way, it's gonna shut stuff down. If you're running away from a lion, you don't need to digest anything. Your immune system does not care anymore. You gotta run. All your energy is going to your heart, your lungs. So those big signals, those big ones, like stress response is a good one. It's like I always kind of bent on domestication biology. It's really cool. You are activating hundreds of things for that activity to come through. This one is just a little same visual, just with more genes. So strictly speaking, this kind of finishes the picture from the last slide, focus right here. Ultimately what these little guys will, sorry, I didn't tap that aggressively. Ultimately what these guys are gonna do, one of them is gonna go activate something called the transcription factor. TF, otherwise known They wait dormant in your nucleus, ready to activate and turn on all kinds of cool genes. They need the signal to turn on. You make that gene into mRNA, you're gonna make it into protein, draw it to 11, and it's good. So a good kind of build off of this is that if an animal has a stressful moment, like we all did just now, we don't necessarily activate genes to survive that. We just kind of open some signal paths and things get kind of ready. But let's say you're running for like an hour from something. You will be activating genes that will then be shutting down your systems you don't need. And they will be activating and pumping out as many genes as you can to sustain the beating that your heart is taking, for example. The gene activation is a lot of the time is the goal with some of these. Not every drug, but it's the most powerful we can do sometimes. So this is pretty much just another example. Yes.
By gene activation, wouldn't that make a permanent change?
Yes, so long as the genes eventually get recycled. So we all have like kind of little recyclers in our cells. So as long as, yeah, once you make a gene, eventually it kind of dies down. That's like, as you kind of come down from a feeling, that's kind of what's happening. Your neurotransmitters come down, other genes kind of come down. So we all have natural mechanisms to like get rid of genes as they like show up too much. So a good example here is a growth factor. Hooray, same thing, it's gonna bind on the top. The receptor, notice, most receptors are gonna have two sides, an opening and a translation down into the cell. And it's not going to do the actual function. It's going to find little secondary messengers. So I'm going to say primary. I'm going to call that the ligand and the receptor. Secondary, and I'm going to say tertiary is that little purple guy right there, activating a gene that you ultimately get. So growth factor, for example, if you accidentally get a cut on something, you got to grow new cells. Your little platelets attract those growth factors, make new genes to make new cells. So again, sort of as a little red text review, one reminder is that every one of your cells, your brain cell, your intestine cell, your heart cell, they carry the genome in a complete copy, complete cookbook for everything you could ever want. Technically, you can take any cell and basically command it to do any sort of gene stuff or any gene that you want in a little dish, given them the right amount of like ingredients. But there's a reason why every one of your cells technically has the same instructions, they just use it differently. Drugs in a lot of cases try to elicit that effect a little more promptly. So again, one last visualization of what genes are. Right. So, good times. You're still stuck in here for another half. But I'm actually maybe gonna do the chalkboard for us. I don't know, I might get us up a little bit. The room is also debilitating. I'm still upset about that. Here's what I can do. Let's start with this though. We can just like kind of be at our tables. So, like we said, let's finish this sugar example. There's a lot of stuff it elicits. So go ahead and get on your phones. Find me pathways that sugar can activate. Think brain, think heart, think lungs, think endocrine, think anything. Good source of this is always going to be follow controversy and weird articles like sugar will make you burn, I don't know, like sugar will make, I don't know. Find a crazy Instagram reel. It's like, eat more sugar. I don't know. But realistically, hopefully you can find like a little life hack. Google image search will always get you the answer faster than the text search because somebody's probably made a fun little overview figure of it. So find some weird stuff that sugar can do. What pathways is that activating and how? Also, I'm not anti-sugar of this. It's a good example. And notice we're in a nice little break for me talking. Hooray.
It's my first thought.
What have anything interesting pop up so far? Right. Yeah, I knew that was coming. Too much sugar will trigger certain things about your pancreas and cancer. Your pancreas doesn't like that much sugar and it kind of gets it angry and debilitates it a bit. So yeah, don't drink the Mountain Dew. I'm gonna throw that away ceremoniously at the end. Let's say PDAC, that is pancreatic cancer abbreviation. Anybody find any brain pathway stuff?
It could block the dopamine receptor, and then it makes you want to eat more sugar to receive the same feeling.
Yeah, right. Hungry. Awesome. Okay. If you block dopamine, it's kind of the opposite of what we just saw with the red text. No more genes being made for that. Oh my God, we need those genes. Better unjam that. Better get more then. Okay. Anything with the heart.
The oxidative or non-oxidative can cause persistent or acute stress.
Yeah. So free sugar is a rare thing in the wild. Like I saw, all the, who's taking biochem? Yeah, that's right. There's all those pathways to break sugar into the fine molecule that we could just have eaten right there, right? If you just give it as the fine molecule, a bunch of the systems are just like, hey, what is this? There's like way too much. And the mitochondria are all like blowing up because they're like, this is too much sugar. So it kind of ruins things and like kind of triggers your mitochondria into going into a, I don't know, they won't be as efficient. They'll kind of burn a bunch of stuff. Another odd theory, I should say oxidative phosphorylation affected. So you guys have heard if you're a cancer patient, you should do keto diet, right? Or like heard that before. Because technically tumors feed very easily off of pure sugar. It's the easiest source. And they can burn out a bunch of lactic acid to do it because that's the non-oxygen pathway and tumors are all like tightly stuffed. There's no oxygen that gets through that. It basically bioengineers the whole environment. All right, we already know, we're already gonna say T2 right there for diabetes. What you can do is you find the things around you everywhere. The way that you can kind of build your confidence sometimes in this class, I'll try to provide examples with stuff. The more that you guys search and find like, oh man, that molecule that's in my Bang energy drink is actually attaching to this receptor and that's doing that. Whoops, those things are awful. Ugh. Guys, small stuff can have a big effect. So, Caitlin brought up blocking dopamine. Did anybody else find any other brain stuff? You have sort of the addictive tendency of it. You see anything else for it? How a molecule can affect brain function? It kind of come through for that. Think about what would happen if you drank that thing. Headache, right? Overwhelming amount of signal. All those activating like genes running around, that's not built into the human genome to activate that much. What would happen to your stomach with that much sugar? Roiling around trying to acidify. So anytime you're thinking about what could this be binding to, think symptoms. All right, little green text story. Who else, anybody played fantasy football, right? Yeah, you know what it is, right? So the weekly like loser about 5 years ago, one of our guys from Colorado, he just got obsessed. He's like, weekly loser should chug a 2-liter Mountain Dew. He was so bad. But anytime anybody didn't do it, they just lose 3 straight games. The curse of the Mountain Dew. It was horrible. So now we call it Mountain Dew. So I think we saw like a YouTube guy like, yeah, I just got a 2-liter Mountain Dew. That's that. So one last little fun thing here. It's kind of an intersection with chemistry and stuff, definitely green text, but who's ever wanted to be a little more risk-taking, right? Yeah, it'd be fun. Toxoplasmosis is a little pathogen. The way that it usually operates is it gets into mice, because mice sadly eat mouse poops, gross. What tox actually does is it mimics the GABA neurotransmitter. GABA calms you. So tox secretes all this toxin that looks like GABA. The mouse is now calm. It doesn't fear anything. Runs right out of its hole and it's like, hey, Mr. Cat. Cat eats it, poops it out. Mice eat the poop. Cycle of life. Plenty of things can affect how your systems in chemistry can be. Diet's the main thing. Most of us don't have this close contact with cats, so it's fine. It's also going to get your dogs if they eat raw meat, so don't let them. That's not mine, but that's okay. Anytime you can affect any of that CNS stuff, central nervous system, I should say, you will elicit some pretty large effects sometimes. So this is just a little like overview, just remember. Everything's the body. There are systems that are built on organs. Those organs are built on tissues. But everything comes down, no matter what, to cell deregulation. One cell at a time can be the absolute issue, and that can be it. You deregulate the entire thing. And it's kind of nuts how many of these things we're making all the time. Most of those are in your gut, because they're all dying and shedding, but at times. Yeah, I mean, we're kind of good here, right? Been a lot of Bio 105, so it's okay. Equally, we're not going to get to cell cycle med until we get to the cancer effectors, where that's where we're going to see how to stop this cycle in cells that are growing way too many times. Follow DNA, yeah. So, last little thing before another break. We have to trick this language. We can do that by destroying receptors. We can mimic ligands. I should write some of these down, sorry. There's plenty of ways. I'm going to say inhibit receptor, okay? You can mimic things that aren't there. Mimic ligand. I think of other creative, fun ways. Equally, too, you can activate plenty of things with drugs. We'll meet those. Those are agonists instead of antagonists. We do have rare drugs that can, remember the little signaling molecules cascade inside? Sometimes you can skip the line and trigger those too. They're rare though. They're hard to get. But this is good. See? This is why we put you through all that orgo, right? Chemistry. Science, Mr. White, right? This is good. We've seen Breaking Bad. Nice job. All right, so we get in I'm hopeful we'll have the time. We'll do some I gotta do the cartoon. We'll do some history on like how it came to be that drugs became the beginning of orgo, basically, and how powerful that was. Here's found another movie scene. Smiling. Betrayal. Brother. Evil. All right, just a break. It's fine. Should have given Scott treats, yeah. All right, 5 minutes. We'll reconvene. That was a lot of red.
Oh, yeah.
Oh, what did you talk about?
So we're gonna do. Oh, did you think I was talking to you?
I guess not.
Next, so we're gonna meet with Kelly next Monday. Whatever. Our events are gonna be, or our meetings are gonna be on Tuesdays. Yeah, maybe not doing Thursday, so, yeah, that'll work. What else? As far as location, we're gonna try get more of like a costume style room, lecture room, because the last room we had, like the whole circle thing, that was, yeah, that was, yeah. Because if you get a lot of speakers there, that's what are you gonna do? Stand in the middle and like twirl? So, yeah, that's kind of the main things.
I have one girl from Manchester. She did her last year session.
Yeah, I have.
Or I mean, probably Zoom.
I've reached out to a lot of people, but they just, we haven't been able to it. So, yeah, we need to have like dates, and then I'm gonna reach out and say, this is one of this, because like. One of the doctors, and really good family friend, I shout out to cardiologist, he was your friend, did love saying, he's from, so like. He's super funny too, so he would be.
You could be like, I don't remember anything on it.
Yeah, right, so that would be good. Another person, who just like on his vascular shit for your residency. You have that, he's like the associate director or whatever. So, interesting perspective. That's a nice job, so. Again, just think it is.
It's okay.
I think it's kind of rude that she asked for.
So it's not really what's the thing. He's one of the ones.
And they're like, yeah, let's do come on. Not like. I was there tonight and then tonight.
They might have come on for that one. They might have come on. And the only thing that might have been there.
She was. I can't like sitting there. I could turn it to. She was ready by Reddit.
I know I can.
I just parked in 3 months. Yeah.
No, thank you. Or you had 5 months. You had daughters?
Yeah,
It's not for, I don't know.
All right, so time for some red text that we haven't seen before. Consider this our Friday class, right? We're moving into the third one. So far, yeah, so far this isn't too bad. Yeah, I don't think it's that bad of a flight so far. We'll see. So, what's a drug? There we go. Couple needs there. Now, obviously, they're posted, so consider noting why each one of these must be. So anytime you see anything that's on the key info and it's already posted, you wouldn't rewrite it, right? Why must it bind? That one's semi-self-intuitive, right? It needs to actually attach to something. It needs to be, you need to be able to do it over and over again. Plenty of times somebody takes a drug one time, does something weird, and take it the other 10 times, nothing happens. Whatever the effect has to be, it has to be consistent. Do you think big signaling binder type things that send signals throughout the bloodstream as a consequence are more reproducible or less? Probably a little less. Those effects are bigger. Got to be able to deliver it. Easiest way is a pill. Okay. There's plenty of others though. We'll meet them, but patients don't like those. Equally, your body metabolizes a lot of stuff. Plenty of things that were hopeful drugs in mice suddenly get into a human digestion system, gone. And like we kind of talked about before, the specificity has to be enough that you can't kill the patient, or at least not too many. There's a line. Next, size, which is the last one and the most easy to break. Why should it be small? Think to yourself. Yeah. In plenty of cases, some of these small enough ones can do it. If your target's inside a cell, it better be small enough, or it better have an avenue in. H2O itself is only $18. Aspirin, yeah, I don't want anything like that. So this is red text because obviously we'll see aspirin. But the cool thing, the reason that's here, is to show that this tiny little David can take down a Goliath. Your enzymes, your genes, are huge. But one soldier in the right place can debilitate an entire, base, basically. You plug that catalytic site of that enzyme, just in that one spot, and it can fit all the way through that thing, binds, the thing's done, spent. Small spaces to fit in those active pockets are very important. It's only red text, obviously we'll see aspirin later, but as long as you can obstruct that section right there, typically going to be something tiny like that. Those pockets built on amino acids of genes are really fine, like really small. So finding that drug that looks just enough in there that's the perfect thing. And aspirin's there's millions of unsuccessful drugs. Aspirin's one of the ones that's like, oh my God, that works so well. So, technically, yeah, we're getting a little weird here. Lipinski's rule of 5. One of the first organic chemists was like, all right, to be a drug, you're probably gonna hit 4 out of these 5. Let's take a look. First one, you wanna be small. Notice that that's the lesser sign right there. Smaller drugs can run and attack their targets much easier. They're more swift. You don't want to have too many, here we go with the ordo. You do not want to have too many centers of chirality in the drug. It's going to spin around too much. It's going to be too weird. You want it to be consistent. ugly, sorry, low chirality is what that says. There it is. The amount of OHs should be small. OHs are extremely reactive. Couldn't get a better overview photo that like have a little more text to it, sadly. OHs are the thing that are constantly being attacked by your body, digested, changed, whatever. So you can't have too many of them. Similar vein here, you cannot have too many electron acceptors or too many electron donors. If you accept too many electrons, you're going to get changed. If you give too many electrons, you're going to touch way too many things in the body, and it's going to hurt something. Antioxidants. There's a reason we drink and eat those, so that you don't have stuff jumping electrons all over your body and screwing things up. I'm going to say, this is a bit of a summary, low reactivity. Don't want something going in there and messing with just anything. Last one, probably the toughest one is you've got to be able to be hydrophilic because you have to exist in the blood of the body. It can't clump up. Some drugs are kind of hydrophobic, they usually don't do very well. Now, somehow on the other end, you have to at least be able to briefly exist. And this is only for some drugs. You have to briefly be able to pass that barrier. Not every drug needs to do that, since a lot of stuff's on the outside, right? I'm going to say, can swim, can cross. There we go, see? We turned some scary chemistry into some fun little phrases. Now, and we've talked about this, plenty of drugs break one of these rules. It's fine. There's plenty of drugs that are bigger. There's plenty of drugs that can't cross. There's plenty of drugs that have a bunch of chiral centers and they somehow are okay. So when you design a drug, a couple ways around this. There we go. These little structures are good. If you know your target protein and what that looks like, perfect. You know what the known ligand of that thing that you're trying to mimic is? Nice. What you can do is take the known structure and just do some computer simulations and dock and see what a similar structure is going to be, turn it into a drug. Now notice, let's actually go to the opposite of this, unknown and unknown. Don't know what this protein looks like. You don't know its ligand. In the purple box, you're going to be starting from square one. You're going to be trying to combine tons of different amino acids that are like that gene, not it. Same thing with the ligand. Notice that if you know the signal or protein itself, you know the protein, for example, you can design a ligand around what the active site looks like. If you don't know the protein, but you know the ligand, you try and basically, again, you're going to be doing like crystallography and crazy stuff to try and fit a known ligand into something that we don't know how to stop it. I will say that the yellow one is the hardest thing to get us. And starting from square one in purple is also hard. So, lots of ways to get what we need. This is just another visualization. It's in the key info. Just threw out a few more good examples that I like. Kind of wanted to put it in. Hence the blue text, though. So, what we got left? What a clinical trial is. We're going to get into insurance. We're getting into the drug industry. I linked up the seminar that the industry side is based off of. I would recommend watching it. It's a Mayo Grand Rounds. I was lucky enough to be there. I was like, dang, that was a good picture of how this works. Do that today, but let's hit up some bases of trials, how this goes. So before a trial can even begin, trial itself is going to be number 4 right here. That's all the stuff we just did right now. We gotta design what something looks like. We need to validate that it literally binds and touches the thing we want it to hit. This is red text, but again, as I hoping to kind of broaden it. So here, let me give some like summaries on each one of these. So I'm gonna say find fit. Next, I'm gonna say modify. I'm gonna say modify top candidates, basically. So once you get a hit out of those millions and thousands, you kind of mess and change with the drug a little bit. See if you can get a little better. Next, go into some animals. Those first 3 steps are what precede a trial. These are preclinical work. Technically, the little cancer tumor cells that I use are a preclinical model. So we first test them on mice to kill the mouse. That's bad. Then we test it on cancer cells, or in some cases with other drugs, different other cells like in a lab. Next, phase 1 through 4. Let's do this. This is good. I'm glad this is red text because I think people skip this too often, but like this is how to like kind of make sense of how this whole process works. We'll find out why this process is so horrific and time consuming. Phase 1 picks up where we left off with the animals. You were gonna get a group of like 50 or 100 or something, you're just gonna give them the drug. And do they have any side effects or do they die? It's just toxicity, that's it. As you can tell, it's highlighted with just the word safety. We don't care if it works on the drug. These are just healthy individuals that want to score about $500 each. Let me tell you, don't do those, okay? Next, phase 2, probably surprisingly the most important one. Phase 2 is where you get a study group. You say, group A, you're gonna get this new drug. Group B, you're gonna get the old drug. This is your first split. This is where we're first measure and see if group A with the new drug does better. Now, plenty of phase 2s look good. Plenty of phase 1s look fine. You may be at this final phase with 3. 3, we really up the ante. We basically, it was times 10. But on small trial, we go big. Now, the latest, most popular trial with COVID vaccines, that reached up to 45,000 in phase 3 in each way. What phase 3 is also called a validation, basically. We saw it in a small population, do it one more time, do it again. So, not to badmouth phase 4, but technically phase 4 is just, you can market it. We're just kind of still tracking the data. So, there's really, the science is kind of done at this point. So, for example, is aspirin a phase 4 trial right now? No, we're done. We're toast. There are plenty of experimental drugs, though, that if you need to take, for example, for yellow fever, dengue fever, technically they're phase 4s. They're good, but you're still part of the study because you want to make sure. So why not? But phase 4 is much more like it's good than a lot of patients will be like, oh no, it's still in phase 4. It's like, you're fine. So probably all right. Okay.
I have a question.
Oh, go ahead. Yeah.
So is there any, I guess, I don't know, as far as like situations where this has happened, but like, let's hypothetically say we're in 4 and like the population you're looking at, there's conflicting evidence.
It starts to build.
Do you move back into phase 3?
If you're responsible. Yeah. But the good news with peer review is once it's phase 4, it's kind of up for everybody to just grab and test.
Okay.
And other voices can come through and say, I did not see that in my patients. That did not work. And then people start talking. I mean, that's a big deal why conferences work. If you find there are people that are like, that drug was trash. You start that conversation and the patients get better for it. So yeah, there's plenty of times, strangely enough, that 4 fails. It shouldn't. And usually what that means is somebody messed up in 2 or 3. So Why do we need all these things too? This is built on history. In the old days, and this is quoting the book, basically all you needed to run a clinical trial was people to just kind of volunteer for it and tell them the dangers, and they're like, I want the money, I don't care. Now we have a lot more safeguards. There's certain things that like you can't get past animal stuff. You're like, no way, we're starting a trial here. In the old days, it was like Wild West and horrible. Needless to say, too, places would poach on poor and vulnerable areas to get a population going. Why we have so many more rules here. Equally, you can't just drug anything you want. You have to validate that we need to do this at all. One of the most popular selling drugs on the planet, Viagra. Did we need to do that? No, but it was a heart medication to start, and it worked pretty well. That's another story. We're kind of getting around that. A lot of needs for these. I'm kind of a lot of text for red. The big idea here is that I'm going to highlight the two that I'm going to say matter the most. Well, these all matter. Dang it. All right. But the big deal is like why we have so many protections for patients is that before no protections and before a centralized system was stepping in, you kind of just do whatever you want, and that's just not feasible. You also don't communicate results well without this. There has to be a reason to do something too. Okay. Equally, recently-ish, all trials must, to move on, demonstrate in 2 or 3 phase, they must demonstrate that there is a significantly meaningful new effect from this drug, or we just yank it. There's no reason to start something new that we don't know about. There's 2 ways to do this. One is the old way, which is, it's not evil, but like, it's not great. Like if your relative has cancer, do you want them being in the trial with placebo? No, that actually doesn't even happen anymore. And move to this model. Now we've had so many drugs, you can use the old one, or you're gonna get the new one. We're gonna measure and see who splits and if there's a meaningful difference. If plenty of trials end here, then both drugs just kind of work fine. But we're gonna go with the old one in that case. Maybe there's gonna be special scenarios. There must be some kind of split to justify any of this, basically. Yeah, in death, what I mean is that can be the thing, like, can we prevent that? We don't want that to happen. So one of the things that during the trials that you have to carefully comb through, you don't need to know each one of these. You just need to know AEs, adverse effects. During trials, we are looking for these. Some of these could be drug related in purple right there. Some can be less related. Or perhaps they're just kind of triggered by the presence of the disease in your drug. Maybe they're not quite your fault. There's different ways to sort of categorize these. Equally, there's plenty of adverse effects that are fine. Like, you got diarrhea, well, all right. Serious adverse effects is like a cytokine storm where your immune system turns on you and kills you. That's serious. So technically through these you have, this is an easier way, but any adverse effect goes through stages 1 and stage 2, they're fine, they're acceptable. But anything, oops, that was done to do it like that. Anything past a 3 and a 4, you got to track that percent and you have to report how many people are suffering these really bad things. Because even the best drug can get pulled right off if 40% of people are having internal bleeding, obviously. You want that safety of that. Yeah, like I said, there's no joke, man. Drugs have weird effects. Don't do a trial if you can help it. You don't know what the brain is responding to at any moment. Okay, so big question that you'll often get, 2 big questions. How did we get this done so quickly? That didn't seem right. Here's the fun deal with this. So many people got COVID in the trial immediately after getting the vaccine that the study was over. COVID was everywhere. Fun times. The exact opposite reason that for certain diseases that are hard to control and study in a single box, like Ebola, and there's not enough, there's never been the raw ability to separate group A treatment, group B treatment, or group C. When you hear about, so like at UNMC in Omaha, they cured a person with Ebola. They have done that multiple times, but they've used every treatment in the book. They send in antiretrovirals, they send in plasma from a cured person, and they do transplant, like marrow transplants and stuff. We don't know which one worked or if they all worked in conjunction somehow. Because when that happens to you, kind of the trial stuff goes a little out the window and you just gotta survive this. So, details are posted because they're helpful to know, I think, about which one. So let's break a couple rules right here and take a look at something that doesn't really fit. Insulin's not an organic molecule, insulin's a gene. Very much a drug though. Breaks every single one of Lipinski's rules though, right? These are biologics and biosimilars. This is a size versus surface area versus weight. So here's all our small guys, right? Small 500, right? A lot of natural products that we can use to mimic drug effects. This is your THCs, your CBDs, stuff that comes out of the ground. Then here, we just make big old proteins and send them in. We can engineer insulin now. Used to have to get it out of pigs. It's very much a drug, but it's not an organic molecule. So biosimilar, biologic is the big broad term of that. And we're not even into cells that we can trigger into becoming actual like fighter, killer, pathogen hunter type stuff. That is not even on the slide. We'll get there in week 15. Got a lot of flights before week 15, huh? Now, here we go. This is rising up. In the beginning, a drug company will R&D anywhere for a single pocket of an enzyme, 10,000, 25,000 just plates of 384 at a time and see what happens and what works. You filter those to the preclinical animal ones. That may only be 20 at most. Again, you'll cross off a bunch from toxicity. You'll cross some more off at phase 1, cross a bunch off at phase 2, even though there's not much left. Phase 3 is kind of that final killer. Now, there's the problem on the bottom. We put a lot of time in this. We're only going to get 10 years out of this. Do you see where the money's starting to get aggressive here? You've got that one drug up there that you probably put in billions of dollars out of the filtering process to get. You're at year 10, you want your money. Other big thing with clinical trials. Sometimes we may find that drugs work and we end up finding out we don't know why. This is the case with most, I would say, most CNS regulatory drugs that go in the brain and try and trigger certain things to happen. We know that they work in certain populations. We don't know why. Sadly, that's going to be the end. A lot of times in current research, we'll try and find why so that you can build better. Brain's a big mystery. A lot of things, a lot of wires crossing that we can't always measure on. So there's a problem here. Research R&D is important. You do need a ton of drugs. This is just kind of green text here. You do need tons of drugs. The greens are biologic drugs. The reds are just normal orgo drugs. But the blue, our friends, is the R&D budgets collectively of the pharmaceutical companies. Are they wrong to do, see where we're having an issue here? There's one clear one that's growing and the others aren't exactly growing. Would it be good if we had a bunch more approved drugs, or is that irresponsible? It's the right way to spend money on things. Here's the other thing. You only got 10 years, it took you 10 years to get to a phase 4, and now you're finally on shelves. Somebody's going to make a generic of you in about 10 years. So you got to squeeze as much money out of that. And equally, there's a couple things you can do to get about 2-3 years out of your patent if you kind of sue the government, but it's more of a little relationship. You kind of say like, hey, we need more time. we are like repatenting this for 2 more years. Okay, so Aiden, you talked about a drug that, hey, it looked fine, and then it wasn't. This all actually happened in Europe. So in Germany, we'll see in the little history slide at the very end why Germany is one of the big chemical giants. They came up with thalidomide because women are hysterical and it's like, let's calm them down. That was medicine about 70 years ago. Sorry, horrible. Turns out there were some reports that thalidomide, if anybody who was on that, would have a pregnancy defect. Horrible, like limbs missing or bunches of stuff. I didn't show any of it because it's like, it's really grisly and I feel bad. So KH here was just somebody in the FDA, growing FDA that didn't have a role. She said, I've collected all this data and I'm neurotic. That drug does not belong in the United States. And those companies came after her hard. Newspaper articles calling her a witch, saying that she's going to kill people by not allowing their drug to go out and help everything. So as hard as bureaucracy can be, still be a big hero for saying, she's the first person to be like, no, I'm standing my ground. You need to prove that it's safe in pregnant people. And they could not produce that proof. More info kept coming out of Germany and Europe. Not available. So it is important to kick stuff out, especially when on the surface it may seem okay. This is also our first look at that. Drugs affect the fetal barrier much differently than they affect just an adult. So everybody always talks about precision medicine. We'll treat every patient with the same thing or with like different stuff, right? There's a problem with that. I want all the money coming in through one drug though. Why would I develop 10 drugs when it took all that time to get just one? I want everybody to be treated with the same thing. That means more money is going to funnel to me. That's one thing standing in the way of a precision approach in medicine, that you could treat every patient very differently. There's not much money in precision. Talk a little bit about it in a sec, but how rare disease drugs, where not a lot of patients are out there to pay for it, are hard to develop. They're gonna be tough to sell. Like we talked about with production. Something goes wrong in the production line, you've lost all of this. Now there's a shortage of a drug. That also shows how closely do we want to tie to private in terms of healthcare. If something goes down with them, problem. Now, I use the word slim profit margins. That's a relative term. But to them, they see it that way. All right, let's take a little break. That was a lot of drug. The next piece that we're going to do, I'm going to answer all your questions about how pharmas are evil and if they are at all, right? So take about 3 minutes. Think to yourself, though, what are your impressions of this?
Yeah, so the you get yourself. Like the most, what,
At this stage of your life, like that's possible. It's so hard
Mine is different.
It went from like very.
This or pen.
Like, this is awful.
Oh, I know, but I'm doing.
You wanna read it?
I went from this.
It's hard because of.
I said that.
Yeah.
All right, so who's ready for this? Conspiracy time. It's time to dish on capitalism too, right? Let's do this. All right, in orange, you've got profit margin high, you got revenue to the right. All those orange ones are farm companies. That's the S&P 500. They ain't hurting, right? There's that. Next, we do have to talk about a few things. They're making more money, but we're prescribing more than ever. Again, like I said, could my dad exercise and walk a little bit instead of having statins? Probably. Tom's not gonna do that. Here's their argument, yeah. That's what it's on. Your parents become your kids when you're 30, you're ready. Okay, here's their argument. We spend so much money, the market's after us. How dare you stifle our innovation and research? Talk about rare drugs or rare disease drugs, right? Well, when rare disease drugs get bought, gotta make some money. There you go. Okay. Unjustifiable prices. You guys have seen these. Plenty of stuff since that, yeah, since what, 10 years ago, we've got plenty of stuff up here that's like, okay, where's this coming from? Can't be a production issue since most of these are actually generics now. Here's all the stuff that's gotten cheaper over the years. Here's the stuff that's gotten expensive. What's that? Oh, that's insulin. Whoops. So the focus here is going to be on insulin improvement. It's the most grievous example of how you price a drug out. Okay. Here's the problem too. And we know that it's cheap to make. We make it in a lab with bacteria. How's this happening to us then? Well, you have people crossing the border in Mexico to buy drugs. It was the same thing that happened in the HIV crisis too. Okay, number one, we'll just do little circles here. Why can we charge so much? Because you're gonna die without it. You got a really sensitive population right there. Number two, Keenan, monopoly. How do you monopolize a gene that we've made for 50 years? Tell you what, Eli Lilly, what is it, AbbVie, and one more have patented insulin. They call it something else. They call it like they're one of their goofy names. But ultimately, they own that. Over patent engineering. So I talked about, right? Insulin's generic by now. Yeah. They will keep naming it and repatenting it and adding a small chemical group that has no meaningful change. New chemical, quote unquote, new drug, new patent. Okay, number four, similar to what we just talked about, planned obsolescence. Our new fancy insulin with a phosphate group in this meaningless spot. Physicians are often encouraged by the companies to, why would you treat your patient with old insulin when we have the new, better product now? I said no difference. So we're crossing into a certain realm here. Not every physician is some money-grubbing, horrible person, but anybody who prescribes has a choice. And they are constantly encouraged to use new. I saw a charge once on one of the physicians at Mayo called honorarium. That's for like speaking or something. It was like $7,000. I was like, wow, I guess that's why it works at the Mayo Clinic. It was from a drug company. Okay. Next. Biosimilars are slow. It's hard to make a gene. It's hard to create your own version of it. You guys have heard maybe of Mark Cuban doing his like drug company, right? Where he's like, screw this, I'm just gonna make this cheap. It's taken a few years to get rolling. This is a mass production thing. You can't just roll a factory out of nothing. 6 is important, it's kind of hidden down here. Here we go. The 3 companies that control the major 3 products of insulin, they say they're competing, but every time one raises the price, the same day the others do. You think that one of them would just go a little cheaper, right? No. You can't legally prove that in court of law, sadly. They're like, I don't know. And trust me, they got really expensive lawyers too. We're gonna get into that in a sec. So anytime that Eli Lilly raises its insulin prices, the others do too. Okay, next, middles. Production line to patient. Who's on the way there? Myelin is one of the insulins right there. Let's take a look. Blur. Okay, number 1, you gotta talk to your little salespeople, and they talk to the insurers, and it's like, buy our stuff. Okay, the insurers are like, we'll insure that. The wholesalers distribute it to places, and the pharmacies retail it. Pharmacies themselves are not really the problem, you can see here in a lot of ways. They are taking a cut of this way before you get to patients. Now, the middlemen are not the largest issue, but let's roll this up like that. The higher the price, the more money everyone makes in here, okay? Company, wholesaler, pharmacy. The higher that price, the bigger the small cut that each one of them takes. I'm not being a conspiracy theorist right now. This is Vince Rajkumar's seminar at Mayo. Watch the whole thing, I promise. So, there's one other thing that's happening here. Patient gets the rebate. Rebate is like sort of a kick on the insurance a little bit. They're not really the ones paying the ton here. It's the uninsured that are going to just get destroyed here. They're the ones with no protection. So, like, that's sort of why ACA did what it did about saying you can't punish my client for having type 1. You have to give them the same insurance that this person has technically. Okay, number 2, where do the rebates go? Log back to the company mostly. Technically, the rebates that the company is sending to the middlemen to cover the cost of the expensive stuff just get eaten up back by them. Fine. Those are keep growing as well. It's a lot of red text. I promise we'll do broad strokes on this with the exam. Like, I'll ask you to tell me a few things about stuff, so don't feel like you need to memorize anything. Now, the hard part with this with middlemen is that they actually are not really the problem. They only are that third right there. And honestly, like, yeah, you do need distribution, you do need a couple things. Like, the business people will tell you, you do need to communicate and talk. Okay, sure. But then there we go. The big guys, right? Lawyers, money, lobbying, it's a real thing. They're stealing our country. Oh my God, and we're in here talking, doing stuff. We should all be poli-sci majors. No, it's fine. That's called econ light. No offense, wait, don't we have a poli-sci major in here? I can't remember. No, that's in genetics, never mind. I like making fun of poli-sci, all my friends at Carl's board. Now, so when the time comes that it's like, oh wow, 90% of the country would like be okay with this? Nah. Okay. And as you can tell, things are getting super expensive, especially one of the most sensitive populations you got out there is cancer. Because there's not, you can't really turn anywhere else, same as insulin, in a different way. Okay, 2 reasons. This might be changing soon. So, most countries can negotiate as a government against the companies and say, well, that's ridiculous, you're paying over there for that. We actually didn't have that power until I literally think a few months ago in one of the bills. Okay. Next thing, the drug companies, when they need to, they'll pay some of that money to make sure the patient doesn't pay too much. Then the patient's happy. They're like, all right, whatever. So, first person silences the government to advocate. Our VA can, Veterans Affairs can. Now I think most of our institutions are gonna be able to also do that. That's why the VA is a lot cheaper, because they can say, you're paying for this in Canada and Mexico this much, we're gonna get at least some of that price. Next, patients, who's ever been in a doctor's office and just felt absolutely alone? Like, I don't know anybody, these people are good, I have no idea what's happening right now. Most patients feel that way. Comes down to the team, and it comes down to whoever's prescribing that drug to make the responsible call here. That choice comes down to physicians. Now, here's a problem. You're not salaried. It depends on how many patients you see, it's how much money you get. Mayo's unique is that they are salaried as physicians. They don't need to rush patients. That's kind of part of their chi. Not only that, if you're getting to see more patients, the more you prescribe, the more kickback you get too. There'll come a moment that if you guys are in control of the pad, you get to kind of decide that. So, like we talked about, hopefully we can negotiate. And hopefully there's a model out there that says value-based price, which says if a drug is truly that good and the effect is that measurable, we'll price it based on the measurement. Gleevac's a cancer drug that works so damn well, it cures people. It should, that would be one that you could actually maybe pay for that. But the others, those effects are tiny. The guy on the right's not a good guy, he actually made a thing that destroyed negotiation. So that's what this bill is. Sorry, we can't negotiate. Our prices are high, we can dictate whatever we want. So if we used Medicare Part D or the VA price, you could look more a little bit like Europe and Canada because those negotiating tables will get the companies down. Right now, there's no negotiating table. They can raise the price whenever they want. So one way to stop this would be to stop the rebates. Guess what the other spot is? We take less money on a prescription. Let's take a look over here. Same drug, if prescribed through CVS and Target, they're not necessarily evil, but they're partaking in sort of the giant middlemen. $900. A little bit of that rebate goes in your pocket as a physician now. Okay? It's not the case everywhere, but it is in most struggling hospitals. If you're a hospital, why not just let the drug companies pay our doctors, right? Guess what? Health Warehouse, Costco, Sam's Clubs. Oops. The little Mark Cuban thing that he's trying to do. I'm not trying to be a math fan or anything, but there are affordable options. The physician chooses. Next time you're ever in a patient office, do advocate for yourself and say, what are all the options for a prescription today? And I'm used to getting that question. And you don't have to go all nuts like hospitals are evil. But in a lot of ways, a lot of the responsibility does fall on the prescribing team. And that choice may be being made for you. So when the time comes, like I said, that you guys have that responsibility, you could choose to be, a little more virtuous. Talk to your patients about affordable options. Costco's always going to be one that's okay. That's just their model. They don't price out. So again, patents again, the reason this is in red text is because the companies will always say, we got to get more money. It's not fair. You're going to make a generic out of us after we put in all the work, right? And they did put in a lot of work, about 10 years to get there. There's a lot of different types of patents too. Do you know there's a totally just plant patent? That's just its own thing. Kind of fun. This is that appeal that I talked about too. Once it expires, you can appeal it with the government. They'll maybe give you 2 more years or so. Oh yeah, 3-year appeals. There you go. Okay. So to their credit, unfortunately, they did put in a lot. They did put in a lot of time. You're probably going to get 5 years of profit out there. I mean, think about it too. You got to market the thing. You got to have the dumb drug commercials where everybody's like running around in a field, right? You got to hire the actors and do the whole thing before people are like, wow, I should really go to my physician and buy that thing for myself. So that's where we're at with why this is complicated. Now, if it can exist in Europe and Canada, fine. There's probably a role here for us to have a better system, to say the least. So go ahead and take a little break in order to make a choice. We're going to talk about insurance or we're gonna do a little fun green text history and just kind of chill. We'll do insurance later. So take a little break, kind of reflect on if anything was surprising in the last 10 minutes that we just did there, or if that was totally expected. There you go.
It's like. This. Really? Yeah. They just asymmetric. No one knew it was. I'll just say everyone
No, I have it on the list though. I have heard it's good, yeah.
Yeah.
Okay, yeah, it's kind of, okay, wait, it's gonna be, yep, it's gonna be, if we can addict our population, even better.
Yeah.
And they knew it was addictive, and they're like, nah. So,
Yeah, it was pretty crazy. I just watched it. I was like,
yeah. I remember, yeah, like in high school, like I got injury, like broke my back and shit, and they're like, here's a bunch of Percocet. And I was like, what do I do with this if I don't want it? And they're like, I don't know, here's another one. So, yeah, that was fun. All right. Insurance is actually pretty quick. We're just gonna cover some of the major stuff. Like, honestly, this is a good little life lesson time. Let's do it. All right. So as we go through insurance, essentially the system is that if an emergency happens, we'll help you so you don't go into massive debt. Because medicine's expensive. So preventative care, big one. Technically, most preventative care is outlined for free in most insurances. This is most apparent in dental insurance, right? You get 2 free cleanings. Makes sense, right? Then the insurer doesn't have to pay for the giant tooth extraction operation, right? They don't want to pay for that, it's a pain. Okay, additional needed care, more care, hooray. Now, obviously, as you get older, your insurance rates can go up because you're unhealthier and you're gonna die statistically. Dang it. So, take a look at some things. To begin, and this is good because some of you are gonna go out and get jobs soon and you're gonna select insurances, right? We're in single parent, so in Minnesota, we only got one plan, so. Now, number one, premium is the month, how much you pay a month to the insurance company. Deductible, another good one to pay attention to. How much will you pay raw out of pocket until we step in as the insurance company? How much are we gonna go? Now here's the little sneak, okay? Co-insurance. What does that mean? Once you meet the deductible, what percent are you still gonna pay? They're still gonna get you a little bit. And then finally, the last feature, maximum out of pocket of this plan is this much. So in a massive car emergency, you know, reattaching an arm type of 200 grand type of something, max forever will be that number. So you guys have probably seen emergency insurance plans where it's like deductibles like $5 a month. That number adds 0 too. So don't get the car wrecked then. So network versus out. Everything sucks if you're out of network. Some people won't even serve you if you're out because it's so expensive, and they are trying to actually be responsible in that case. Sorry about the EEs and stuff. That's like sorry, I can point over there. I should point here. But single is EE. These are when you talk about dependence. That's that word, Dependent could be your spouse, if you're the one buying insurance, or it could be all your children. I tried to do my taxes 1 year once at Mayo, and I said I had 7 dependents. I didn't pay a single dime in taxes. I committed fraud accidentally, and I had to pay it all back in March. Yeah, Emma was like, what the shit are you thinking? 7 dependents? And I was like, I don't know, I just added some numbers up on the sheet. 7 Pomeranians. Like, yeah. So our tax person was like, what? You didn't pay a single dime? I was like, I'm getting paid, baby. This is great. And then they're like, you gotta, oh, a lot of that's coming back. I'm lucky I didn't get in some trouble there. So single versus public. It depends on who does something. Now, technically there's a model where you would just literally pay and say like, all right, I gotta get this, one of the little moles that I got sunburn on, I gotta get that lopped off. I'm just gonna pay you directly for it. That's one model. That's kind of the orange there. Technically, that can get a little out of hand, right? Because one hospital will be like, all right, that'll be like $23,000. Our laser's expensive. That's why you kind of usually have some middle in there that can negotiate a little bit on your behalf, although the government or the insurance is going to be tough. That system is working itself out. We talked about this, single payer healthcare now. Kind of. This is a lot for red text. Let's cross this into a blue. Sorry. Okay. This is blue text. So what you're also looking at a little bit, different plans have different flexibility with their network. HMO is very low premium and cheap, but you better be using that hospital and that referral because they're in our system. We maintain the cheapness by being very specific. If you do a giant plan, you're paying more, and you can go to any hospital and see anyone you want, but you will be paying more every month. So if you're like younger and healthy and everything's fine, a lot of people do HMO because it's like, I'll just go to that doctor and do the referrals from them and we're done. You need more flexibility, like, you have kids or something, and it's like they're all, breaking their legs in sports and stuff. You probably got to pay up for the possibility you're going to see that specialist somewhere out there that may not be your network. So there's a couple other plans. Kind of talked about the emergency plans. Some of those are here. So when the time comes, this is where people get really destroyed by surprise, is when, no, I thought I knew my co-insurance and my max out of pocket. A lot of that stuff goes out the window when the network is out. So be very careful ever doing anything out of network or like seeing an insurance plan that has 0 area hospitals in network, because you will be paying way more than what looks on that thing. So those hospitals they're not getting paid by the insurance on your behalf then. They're going to charge you for their services. Okay, this is just a giant comparison. This is a green text that just reference basically. So you can see why it confuses people, right? And patients especially. So being able to like kind of deftly walk your way through these is always a good way to like be a better healthcare person. But anything on an exam is only going to be those 4 factors that we kind of talked about up here. I will compare plans with you and I will ask you, what's the best thing for this type of patient? Different plans here is going to be different if it's like young, I don't have any problems, I want to pay cheap, versus somebody who's like, I have a chronic condition, I need drugs, right? Going to be differences for patients. All right, green text avoid. We've almost done it. The last 9 minutes. I'm going to tell you some fun stories. It's all green. So put them down. No more notes. You're okay. You made it almost. But I have to have my servant first. So first ever drug we ever find. Inedible plants, meaning they weren't eating them. They were chewing on them or using them as a drug in some case. One of the coolest things is that these are areca nuts in these caves, the first things we ever found as a drug. We think that it's because when animals are in pain, they chew on those things. And we as Homo sapiens saw that, mimicked it. Next evidence is Mesopotamia, Egypt, herbalists. It's the beginning of apothecaries. This is still in plant land though, with what a drug is. And very risky, a lot of death. Number 2, one of the big ones, the first drug wars occurred because peoples that are not from the equator have no genetic resistance to malaria. Your ass dies, because it will kill you. Anywhere that there's hot enough to have malaria and mosquitoes, you're gonna die. Unless people's from this area immune to the most horrible disease on the planet. So anytime you see sickle cell as like, oh, it's a disease, no, doc, that's an immunity. There's a price with that immunity, and it's very rare that it comes so clean, like with one change like this. But that shows how powerful malaria is as a selector. Okay, number one thing we kind of saw, malaria is in South America too. They don't have the same genetic distinctions as people's from right there. They had something different. We found that in the highlands of Peru, Chile, in this area, there was a strange tree that the locals chewed on when they were sick. Got rid of all the symptoms and everything they were looking at. This tree itself is called quinone. We know this today as the derivative is hydroxychloroquinone. What this does to the malaria parasite actually affects your blood cells, causes your little heme groups to bunch up. Parasite doesn't like that, can't eat anymore, dies. It's awesome. This is in red text, but we'll see it later in week like 13 with anti-parasites. So quinone bark, hydroxychloroquine, all the Aaron Rodgers stuff, that's all technically from a tree bark. Wars were fought over that tree bark. The Dutch had massive plantings. They started to control it because you couldn't go in any of the areas in the middle of the world without a bunch of quinone running around or you're dying. Japanese in World War II, they owned most of the quinone in the Pacific in that first phase of the war. A lot of Allied troops died from malaria with no treatment. And the Japanese dropped flyers that said, if you take your medication for malaria that the US gives you, your privates will shrink. And so nobody took their medication and they all died. So now we first started getting away from plants. Organic chemistry begins and the bane of all student biomed existence. But how do we get here? Why are we selling this as like some cure and a hilarious snake oil right there? Eventually, we were able to sort of semi-synthesize quinone's derivatives. So we didn't need the bark anymore. Orgo figured it out for us. Other initial time that we needed chemicals was the Irish potato blight. Every single crop looked like that horrible thing in there. And our first attempts at chemical treatments were rough. You know what? Helped the potato blight? We sprayed them with arsenic. Cute. Poor Irish can't catch a break. Next big one that happened. This is where chemistry really takes off. World War I. World War II gets a lot more press because it's more of a good and evil story. World War I was way damn worse. So trench warfare, living in mud and shit and rats and rotting corpses and stuff. The lines weren't moving. Germany made a decision. They knew that they would lose the war unless they could win in the next few years. Einstein was at the same institution, Max Planck Institution, when they demoed on a pigeon chlorine gas the first time. And the thing rioted and died in some horrible way. Einstein left. This was before the Nazis found power and everything. He saw that and he said, no way. Here's how this works. Water in our system will react to chlorine. It will make multiple chemicals, one semi-harmless, but two reactive species. See those? They'll bind to everything and just burn you from the inside. Before nuclear weapons were here, nothing could end the world. The first time that people saw gas be used on people, that's the first time they're like, oh my God, this could be it. It's a big reason why a lot of the chemical big ones exist in Germany too. It's a long story to get to medication. This is a GIF or GIF, if you're wrong, that melts and shows what it's like to have that much acidity. And it'll melt steel. You can better believe it melts intestines and like lungs and any innards. So this is linked on there, but chlorine gas passes very quickly with the wind. And to this day, we still run tests and make sure that we would be prepared if that happened. So, a little quote here. First time the Germans ever used the gas. They knew that they needed to win the war, they would lose. So they made a deal with the devil and they used it. This quote's from a perspective of a German soldier. as he's walking, and for the first time they cleared this trench, the horrible part that like the animals had come up from the ground, the rats, and it all just died. Just piles of the insects were dead, all the horses. They found all the people on the French side had clawed their throats out or shot themselves because of the pain. And one of the auxiliary Indian soldiers said to himself as he saw the gas coming, he's like, this is the way the world ends. This is how, if London gets bombed with this, or if the other cities, it's an end. And chemistry was the first time that before this, war was this honorable thing. We're gonna march off, we're gonna do our right thing. No, man. We're insects, was the quote that finishes there. It was the first time that war was like, this is horrible. This is hell. It was not honor. Now, chemical companies are sponsored by the governments because that's your nukes, that's your way to win. They need to exist now. So get pests, get rid of them. You have to be clean. We can't have disease. Chemical giants that were just poisoning humans were like, nah, let's poison bugs. You need to be clean. For some right reason too. And typhus spread from the trenches all around the world. There's certain theories that civilization would have ended with how many people would have died, with how many concentrated people were in one spot and so disgusting and spreading back. So there is a rule for killing mosquitoes with that many chemicals. There was one thing that happened. Chemicals still had sort of a very dark cycle that somewhat continues today. Didn't work on plants, try it on insects. Still too toxic. Versailles Treaty really punished Germany for a lot of ways. And if they wanted to ramp things up, they had to do things in secret. Cyclone is a gas that was used to try and kill mosquitoes. The German word for cyclone is Zyklon. The original version of that was a pesticide. They named it something called Zyklon version B. That's the gas that was used in the concentration camps. No, sorry. Chambers like that. And it takes the whole chemical side of this to a whole new, horrifying level of where that led. Here's the other thing. In this, besides Zyklon, you know the Nazis actually made all these nerve gases right here, and they had them the whole time. Only reason they never dropped it on London, they thought the Allies had them too. And they're like, if we drop it on London, we're gonna get dropped too. We did not have these. So they could have ended the war in a lot of very dark ways. To this day, sarin is one of the things that we still have to work around. Normally, acetylcholine that binds between muscles causes contraction. So that gets broken down. With sarin, no breakdown. You've seen people die from nerve gases where they just convulse. It's because the muscles don't stop moving and twitching. Heart goes down, lungs go down, everything goes down. So, where these all came through, the chemical giants needed a way to survive. We also get into bugs. We'll probably finish this conversation later because this flight's been long. So, we did it. All right, good job.